Welcome to AP Biology. Today's presentation is going to be on cancer. So there's two types of genes involved in cancer. One of them is a proto-oncogene, and the other one's called a tumor suppressor gene. So there's lots of proto-oncogenes. The ones we talked about in the previous video were RAS, RAF, MAC, ERK. There was a bunch of them. If you look at the signal for normal mitosis, a growth factor is going to bind to the receptor tyrosine kinase receptor, and it's going to cause a cascade. And during this phosphorylation cascade, there's lots of involved enzymes. So we have SOS and RAS and RAF and MAC and ERK. And a mutation in any one of these enzymes to put it into its phosphorylated stage permanently will cause this chain to permanently turn on. In other words, the cell's nucleus will constantly get a signal that it's time to divide. Part of cancer is having a mutated proto-oncogene to tell the cell to always have a signal go into the cell that it's time to divide. Another part of cancer is to have a modified tumor suppressor gene so that your checkpoint that normally stops the process and double checks DNA for damage and all those are turned off. So here's our checkpoints. Normally, when a cell gets a signal it's time to divide, a gene called P53 will go through and check the DNA and make sure it's all intact and make sure the cell's big enough and make sure all the stuff's going on that we're ready to divide. But if you have a mutated P53 at this site, then it doesn't work. And if your mutated P53 doesn't work, then the cell will blow right through this checkpoint before checking the DNA for any damage or anything, and we will replicate damaged DNA. When you do that, you get mutated cells. So these mutations accumulate over time, and they tend to create cells, at least some cells, that will break free of the normal constraints that cells are under. There are other checkpoints at the end of G2 and during M that are also very important as well. And if you have a mutated form at this G2, that's called BRCA. It was the first two genes that we discovered that are related to breast cancer. The BR stands for breast and the CA stands for cancer. So BRCA1 and BRCA2 are checkpoint related molecules, and if you have mutated forms of those, just for example, I'll give you a normal situation for a female is about 1 in 8, 1 in 9 females will develop breast cancer sometime in their life. If you have a mutated BRCA gene, your risk goes up to 90%. So instead of being 12% or so, it goes up to 90%, which is a huge increase. And a lot of those women with mutated BRCA genes will get cancer in their 30s and 40s. So, cancer is basically uncontrolled cell division. Okay, so we have a normal gene that helps us stop the mutation of defective DNA, but when that gene gets mutated, then it allows us to copy mutated DNA, which eventually can lead to a lot of bad things. So normally P53, if there's damage, it will shut things down, okay? Or it will correct the damage and you'll get normal cell production. But if you can't correct the damage because you got a bad P53, now your mutated or damaged form of DNA is going to get passed on and can eventually sometimes lead to cancer cells. It doesn't always lead to cancer cells, but sometimes it does. So a few of the things that have to happen in order for a tumor to be really successful, not that we want them to be successful, but for a tumor to be successful and cause significant cancer, They have to have unlimited growth turned on. So there's your proto-oncogene has to be turned on. You have to turn off P53. Every single tumor in everyone's body has a P53 that's turned off. That will also allow it to escape apoptosis, which is kind of a programmed cell death if things are going wrong. And for immortality to happen, you have to have other genes that are modified. You need modified genes to increase blood flow to the tumor. And in order to get over the anchor, see, cells normally want to be anchored and they want to grow in a single layer. We call it density dependence. And to get over those two things, the fact that we want to be anchored and grow in a single layer, we have to have modifications to those genes. And so in order to grow and form a tumor and break free from that tumor and spread to other parts of the body, you have to have modified genes. So lots of genes have to be modified in order to get to a stage where you have a really bad tumor. You can see here it talks about mutations in tumor suppressor genes can start. Mutations in oncogenes can really promote the growth. More mutations in your tumor suppressors can cause even faster growth. You get additional mutations that are going to cause the cell to break free from its encapsulated state and start spreading to other parts of the body. So it's just a constant kind of continuation of mutations that causes this cancer to become more and more harmful. What types of things are going to cause cancer that we may be exposed to on a regular basis? Well, we know UV radiation. We always talk about skin cancer. Chemicals that people work with, herbicides and fungicides and insecticides and different chemicals people work with all the time have the potential risk of causing cancer. Being exposed to certain types of radiation like X-ray radiation and gamma radiation and things that are involved in like nuclear medicine and nuclear energy. Those radiations can be pretty harmful. There are pollutants that come out of our cars and out of our buildings and out of our power plants that can cause cancer. Radon comes out of the ground. That can cause cancer. Cigarette smoking and smoking pretty much everything you smoke, if you're putting those chemicals into your body, those can cause cancer. Age, the older you get, the more risk there is of a mutation, an accumulation of mutations happening. So age is naturally going to cause more cancer. Genetics, if you're born with bad tumor suppressors or bad proto-oncogenes, then that can potentially cause an increased risk of cancer. But the crazy thing is, most of the time, when people get cancer, it's just bad luck. They happen to have a mutation in an important spot that causes a change that leads to the development of cancer. So we can do a lot of things to prevent all this stuff, and that's going to reduce our risk. We can reduce our risk maybe by 40, 50%, but bad luck's going to play a role. Even if some people do everything right, bad luck can play a role and just cause you to get cancer anyway. So benign tumors aren't very harmful. Malignant tumors are very harmful. Right now, we discover most tumors in stage 3, 4, or 5 because that's when they get a little size to them, and that's when they start causing symptoms, start causing discomfort or whatever. And so that's not good though, because when you get to the malignant stages, the cancers are already to a really harmful stage. We can prevent a few types of cancer, like skin cancer, by constantly observing our bodies, colon cancer by getting colonoscopies. We can catch those at earlier stages, breast cancer with mammograms. But we don't have a way of testing our bodies yet to identify like early lung cancer or liver cancer or pancreatic cancer. But there are some blood tests that are coming out, and they're doing studies with them right now. And if they pan out, we will in the near future be able to test our bodies for different types of cancer just through a blood test. And that will significantly reduce the amount of people that die from cancer, because if you can identify cancer in the benign stage and remove it, you're right back to stage 0. Malignant stage cancer is much more harmful. If we can identify everybody's cancer at this stage, the number of people that are going to die from cancer is going to be insignificant. So as those tests get developed and become better and better, I think the number of people that are going to die from cancer over the next 10 to 20 years is going to be significantly reduced, pushed down to near like maybe 5% of what our current situation is. The traditional treatments for cancer are high energy radiation at localized sites to just kill cells, chemotherapies that kill actively dividing cells. The problem with some chemotherapies is they actually kill all of your actively dividing cells, and some of our actively dividing cells are doing that for a reason. For example, our skin cells actively divide because we need to replace cells on a regular basis. Our blood cells are actively dividing because we lose about 1/100th of our red blood cells every day. Our lining of our digestive tract is actively evolving or actively reproducing, and so that's affected by cancer. Hair is constantly growing, and fingernails are constantly growing. Those things are affected by chemotherapy. And so there's lots of things, your digestive tract, your skin, your blood. People who are doing chemo can become anemic. A lot of times they'll lose their hair because the drugs attack fast dividing cells. Some of the newer drugs, we call them miracle drugs because they can pinpoint certain types of cancer, just go after specific receptors involved in the cancer production process. The first one that we found was Gleevec, and it basically stopped people from getting a certain type of leukemia. It's also been useful in slowing the growth of certain types of cancers in our digestive tract we call GIST. So this was the very first successful kind of miracle drug. There have been other drugs similar to Gleevec that are on the market now for other types of cancers, and they have been pretty successful. But the real savior is going to be when we can identify cancer at early stages so we can treat cancers that are in the stage 1 and 2 instead of stage 3, 4, and 5.